We are covering the process of gene expression. We've just gone through transcription where we have made our messenger RNA. Now this process is taking place in bacteria. Then as soon as the messenger RNA begins to extend off of the DNA, translation can start. However, in eukaryotes, transcription is happening in the nucleus, and the messenger RNA has to move from the nucleus to the ribosomes in the cytosol. So what we actually produce is something called pre-mRNA, and that pre-mRNA has to be processed a little bit before it can be transferred out to the ribosome where translation can begin. For eukaryotes, the final product of transcription is actually what we call a pre-messenger RNA. And that pre-messenger RNA is going to have to be modified a little bit before it is a mature RNA. A mature RNA is an RNA that can be taken to the ribosome and used to produce an amino acid sequence. So what we ultimately want is this messenger RNA sequence that has an untranslated region. Now, this untranslated region is going to be important in ribosome binding. So it's there, it's not going to be part of the final protein, or it's not going to code for parts of the final protein, but it is there for structural reasons. Within that untranslated region, there's a region called the Shine-Dalgarno sequence. Now, the Shine-Dalgarno sequence is important for ribosome binding. This is basically where the ribosome binds and where the 5' end of the messenger RNA is going to bind so that it is in the right position for the ribosome to start at our start codon or our starting point. After that, we have the start codon, which is the signal to start translation. And then we have our protein coding region. This protein coding region is what is going to code for the amino acid. So we're going to use what we call codons to produce our amino acid sequence. At the end of that sequence is a stop codon, which tells the ribosome to stop producing the amino acid. And then we have another untranslated region at the 3' end, and that one is also going to be important for helping the ribosome bind to the messenger RNA, maintaining stability of the messenger RNA in the cytosol. So we'll talk about what these different regions do in a little bit more detail, but this is our goal, to get to this protein coding region with our end caps basically there for stability and getting the ribosome in position. In bacteria, our genes are relatively simply organized. They have coding sequence, they have a start region, they have a stop region, and everything in between gets made into an amino acid sequence. In eukaryotes, though, they're a little bit more complex. In the middle of our coding sequences in eukaryotes, we have regions called introns. Now, introns are non-coding regions. Non-coding means they won't be part of the mature messenger RNA. And so they have to be removed. So the sequences that are going to be removed from the messenger RNA are called introns, and the regions that are going to stay and become part of the mature mRNA are called the exons. So both are transcribed. They both come in the DNA sequence and then are transcribed into the RNA sequence, but those introns are later removed from the sequence. Now, you can remember these by the introns stay in the nucleus because they are removed and degraded. The exons exit the nucleus in the mature mRNA. So that's how you can remember those. So your exons are coding regions because they are going to be part of the code that makes the amino acid sequence. So the number of introns and the size of the introns vary from gene to gene. Some genes have a few introns, others have more introns, no matter what, no matter how many there are, they're going to be removed from the messenger RNA, and the exons are going to be ligated together. We call that splicing, and we'll talk about that in a few minutes. But if we look at two examples here, we've got the ovalbumin gene and the cytochrome B gene. Our exons are the dark blue regions here, and our introns are the light blue in between. And if we look at the mature mRNA down below, we've removed those light blue regions and spliced together the dark blue regions, which are the coding regions. So this process is called mRNA splicing. And splicing just basically means to stick together. So before our mRNA can leave the nucleus, there are three different things that have to happen to it, three different ways that they're processed. The first one is called the 5' cap. So on our messenger RNA, we are going to stick a single molecule, it's a big molecule, that is essentially a modified nucleotide. And this is going to act as an end cap for our messenger RNA. So if we look at this sequence down, or sorry, this molecule down on the bottom, we have this molecule that is essentially a modified guanine. So we've got a guanine here, we've got an extra methyl group, and we've got these 3 phosphates. And then we have the messenger RNA attached to it. And then we also have some other functional groups that are added on. So this is just kind of a very highly modified region of our molecule. Now the reason for this, there are several reasons for this. One is that this is going to help with initiation of translation. The 5' cap is going to help the ribosome come together with the messenger RNA at the proper place. So it's going to help sort of line everything up. And then it also increases the stability of the messenger RNA, because the messenger RNA is probably going to get degraded eventually in the cell. Messenger RNA doesn't hang around very long, and as soon as the cell doesn't need it anymore, it degrades it. So the cap is sort of a way to provide some stability for that messenger RNA molecule. Our next modification is called the 3' poly A tail. Poly A means polyadenylation. So in a polyadenylated tail, the cell tacks on at least 50, sometimes up to 250 adenine nucleotides to the 3' end. Now the main purpose of this is stability of the molecule and protection against degradation. I mentioned with the previous slide that messenger RNA molecules are degraded rather quickly in the cell. They're used until they're not needed anymore, and then in order to reduce the expression of the gene they were made from, they are degraded, and then ribonucleotides can be taken back into the nucleus and rebuilt into new messenger RNA. So there are exonucleases that degrade these messenger RNA molecules, but if a messenger RNA molecule has a long poly A tail, then it's going to be able to continue to be used. So the cell puts this poly A tail on the end so that it can be taken to the ribosome through the cytosol without being degraded enough so that it cannot be translated. Our last bit of processing is the exon splicing that we talked about just a few minutes ago. In exon splicing, the introns are removed and the exons are linked together. Now, they are sort of tagged by consensus sequences at the ends of either of the introns. So there are consensus sequences that code for the splice site or the cut site within our coding region. So as the cell encounters a splice site based on one of these consensus sequences at either end, an exonuclease is going to cut the RNA, going to cut the RNA, and then that intron is going to fall out. When that happens, that intron has a little A in the center called a branch point. And what this A is going to do is basically be a place where this molecule can roll up on itself, and this is called a lariat. Not super important for the rest of our class, but this is how the cell knows that that's an intron and it can be degraded. So what's left are the exons. The introns are removed by cutting them at these splice sites. And then a large molecule enzyme, really called a spliceosome, works to pull the exons together. So this whole process is happening inside of this spliceosome. And basically what it does is holds on to the exons on either side of the splice site, allows the cuts to happen from another region of the molecule, and then ligates or forms a bond between the two exons. Down here on the bottom, we've got that little picture of our intron forming that lariat. And then once the lariat is identified, it can be broken down. So we've got our intron here, 2 exons. The exon is removed, one from exon 1, one from exon 2, and then our exons are spliced together and the intron can be degraded. So you might ask what the point of this is. Why is there even sequence there if we're just going to cut it out? Well, the reason for this sequence is that sometimes our introns and exons can be alternately put together. So in one version of a gene, you might have an intron behaving as an exon, and in another version it might be behaving as an intron. And so what we get is something called alternative processing or alternative splicing. So alternative splicing allows for multiple types of proteins to be produced from the same messenger RNA. And this is accomplished by changing the order or changing the type of exons that are included in the final protein. Now, this really doesn't happen in eukaryotes. It does not happen in prokaryotes, but it does happen in quite a few eukaryote genes. So there's some examples down at the bottom. These are just schematics of how this might happen. But you can see different versions. We've got 3 exons linked together. We've got only 2 exons linked together, and so on in different combinations. And this is going to essentially allow some flexibility in the type of proteins that can be produced from a single messenger RNA.